Thanks for the introduction. Thank you, Dolores. Thank you to the Society for having me to talk here. And I'm pleased to discuss some of my phylogenetic results for these groups of flies. So the trees I'm presenting are, I think, very robust and exciting, but we still are adding some additional taxa, and we're doing some additional analyses. So I'm hoping that the discussion section of this, the conversation, will be helpful and enlightening. Okay. So as Dolores mentioned, I'm a postdoc working on flies. So I'm very interested in the overall evolution of flies, understanding where they came from, particularly ones that affect human society in terms of urban and agricultural pests. Where do those come from? Why are they causing so much damage? What were the evolutionary shifts that led to their promulgation? I'm going to talk about some groups of schizophoran flies. So this is generally broken into two non-monophyletic groups, the calyptrates and the acalyptrates, where the calyptrates are placed within the acalyptrates. And the three most well-known groups in there are Drosophila and the other palmist fruit flies, the tephritid agricultural fruit flies, and the calyptrate that include a lot of veterinary and forensically important flies. This group has had relatively little phylogenetic study because it's extremely complicated. And there's about 40 major lineages that haven't been resolved in terms of their sister group relationships. So that's a major reason why we've been delving into this very large radiation of flies. Traditionally in the acalyptrates, there's about 10 superfamilies, and 5 of those superfamilies are generally resolved as monophyletic in previous studies, and the other 5 are usually paraphyletic or polyphyletic. So we'll be talking today mostly about the nereoids and tephrotoids, which are close relatives in a novel clade that we've identified, and the sphaeroceroids. So overall, I want to go into these two major groups of flies, particularly in reference to how many times and in what circumstances plant feeding has evolved, because those are the two major groups of these acalyptrate flies that first evolved phytophagy as a strategy, where thousands of species of these flies can be attacking living plant hosts. So I'll talk about how we get the data, the sphaeroceroids, and particularly an unusual group of sphaeroceroids that specialize on ferns. Then the modified oboscat clade, which includes tephrotoids, and then delve into the most diverse family of acalyptrates, which is the Tephrididae, which we are finding is a very complicated question in terms of its monophyly. I'm using 3 different major sequencing types: transcriptomes, which is we sequence all the expressed RNA in the organism or the tissue, including some sequences we've sequenced ourselves through an NSF grant or one kite, and a lot of sequences that are already present on NCBI and that I'm sequencing new for these projects. Full genomes, but generally assembled in a non-complete and not annotated. So in other words, just kind of getting major contigs together so that we can put them in the alignment, including some other studies that are churning out data right now. And then anchored hybrid enrichment, which is a probe system where it's a reduced representation exon capture, where we're sequencing about 450 protein coding genes that are single copy, and many of which have been previously informative in Sanger-based studies like CAD and ATS. So transcriptomes, you need to collect them fresh in RNA later. Genomes, you get a lot of data, but it can be difficult to sift through. And anchored hybrid enrichment, you get less data per taxon, but it's a higher, you get more DNA per dollar. So in the lab, we have to collect and identify them, which is a major challenge for these groups of flies. There's thousands and thousands of species and relatively few experts and resources for identification, apart from the pest lineages. And then we get the reads and assemble them, filter them for contamination, and align them with a number of different filtering steps. So the orthology detection, I have about 3,325 gene set where those are single copy protein coding genes within the well-studied, well-annotated reference genomes like Glossina and Drosophila. We align them and then remove outliers, try to do some realignments, and then mask some of the genes to remove potentially poorly aligned regions. And another major part of this is that I tend to like to go back in and check how the alignment algorithms have done. And I think that this alignment step is a major problem in a lot of phylogenomic studies. So let me jump into the taxa, into some of the questions that I'm interested in studying. Earlier this year, we published a major transcriptome-based phylogeny of these acalyptrate flies with 70 taxa. And one of the major findings we found is that a group of siamyzoid flies are sister to the rest with very strong support and relatively long branch lengths. And so the real radiation of the Schizophrena occurs in the group sister to the siamyzoids. And the first two branching lineages within those are the sphaeroceroids and this modified ovoscap clade that includes agricultural fruit flies and relatives. So I'm going to go into the sphaeroceroids. It's quite a diverse and relatively poorly studied group. The two major families in the sphaeroceroids are the heliomyzids, which have 15 subfamilies and an incredible morphological diversity. One group of them is phytophagous, but the rest are on fungus or rotting material. So even people who don't study flies very well can see that there's a huge diversity of morphological syndromes within heliomyzidae, and the wing, head, genitalia character systems are really diverse within the family. So there's no single synapomorphy, and many authors in the past have suggested that it's not monophyletic. The sphaerocerids are probably going to be one of the most diverse groups of flies, but they're very small and relatively poorly studied. You have to collect them in specialized ways often. So what we wanted to do was to investigate the phylogeny of this superfamily, which has never been done before with molecular characters, and trying to sample as thoroughly as we can the major lineages, often representative by family level groups in the classification. Well, that's not always perfect. And I'm doing that with a number of collaborators from around the world. This is our first pass at the phylogeny of sphaeroceroids. And what we find is that the monophyletic superfamily itself is not very clear, but certainly there's four main families that are within the superfamily. And I was able to combine transcriptome, genome, and anchored hybrid enrichment sequences from different taxa and get a overall strong alignment and robust phylogenetic hypothesis. So the two early diverging lineages sister to the rest of the sphaeroceroids are the rhinotaurines and the teratomizids. So rhinotaurines, those are the pictures of the flies around. But by the way, I always really like to put a lot of beautiful fly pictures peppered throughout my talk. So I hope you can enjoy those as we go along. So these are quite unusual, bizarre looking flies, and their life history is also very unclear because they've been reared from living trees that are damaged by beetles. So it's unclear if they're saprophagous, predatory, or phytophagous. What the larvae are doing in those trees is not very clear. And then the heliomyzids, the other major group, are not monophyletic with respect to the sphaerocerids. So this major radiation of saprophagous flies, particularly in tropical areas, is derived from within the heliomyzid grade. One thing I wanted to bring up in particular with this is questions about taxon sampling. So this, I think, is a major limitation in a lot of phylogenetic studies, particularly in insects, where there's so many lineages, many of which are very difficult to come across due to specialized life histories or narrow geographic ranges. So just as a little example, there's a lineage of heliomyzids called the Nemaspathidines that are found in Australia and Chile and southern South America. So when I did not include those Nemaspathidines, the sphaerocerids come out as sister to the calyptrates, very distantly related. But when I do include those Nemaspathidines from the alignment step all the way through, the sphaerocerids are very clearly nested within the heliomyzids. So I'd like to investigate that a little further with some tests, and I suspect it might be something simple like GC bias, but it still shows that including more taxa in your tree can really have strong effects that are not predictable beforehand. These groups, there's a lot of interchange between Australia and temperate South America, back and forth with several different lineages of cerasauroids that have sister groups in Australia or temperate South America, whereas the sphaerosaurids are cosmopolitan and diverse worldwide. The other quite unusual thing about the group is there's no, the sister group to all other acalyptrates, the siamyzoids, doesn't have phytophagous flies, but there's two major lineages in this group that are plant associated, along with several different groups that are predatory, and mostly they're saprophagous. So I want to delve into a bit those fern-feeding teratotomyzid flies. Ferns are an unusual host association for a lot of insects. There's not many insect lineages that really specialize in ferns, but in Eastern Australia, it's quite a diverse region in terms of lower and higher diversity of ferns. So the teratotomyzids are the largest group of flies that specialize in ferns, and they have a lot of strange morphological autapomorphies that have led to their placement being unclear, but I think we can now, with some confidence, suggest that they're part of the sphaerosauroids. And in Australia, there's a huge amount of undescribed species of these fern flies. So I know of at least 30 species in just one genus of the fern flies undescribed throughout Australia. And as you can see, some of these rainforest habitats have a really diverse amount of potential host substrates for the fern flies. And different fern lineages will have specialized fern flies within them. So overall, what I'd like you to take away from here is that within the Schizophrena, we have identified 5 principal lineages, one of which is the sphaerosauroids, which are probably sister to all the Schizophrena, exclusive of siamyzoids. And plant feeding has evolved just twice in the sphaerosauroids, once in the suiliines that attack roots and tubers, and then a second time in the teratotomyzids, but it's quite a unusual and unique plant association. And we're continuing to sample these sphaerosauroids, and hopefully I'll have a nearly complete picture of their higher level phylogeny. The second major lineage within the acalyptrates I'll talk about is this modified ovoscap clade. So our paper we published earlier this year was the first to unite these lineages. But what's kind of surprising about that is the major question about where the tephridids, drosophilids, and calyptrates go has long been kind of unsettled within the phylogeny of acalyptrate flies. So if you look at recent published papers, just that 3 taxon statement can really be all over the place. So right now, the evidence is leaning towards the idea that the tephridids are sister to the drosophilids and calyptrates, but major genomic or ecological studies that seek to compare these groups, it's not a very easy question. But what we found with our transcriptome-based phylogenies is that the tephrotoids are within this modified ovoscap clade. And we went on to sample the tephrotoids more thoroughly with including anchored hybrid enrichment and genome data. Within that modified ovoscap clade, nearly all the families have, I'll show a picture of it in a second, the end of the female abdomen is enlarged and specialized, presumably for laying eggs in different substrates. So no flies have true ovipositors. They only have these an ovoscap for laying eggs. But in this group, they're often enlarged and armored and have sclerites or setae. Most of these lineages are saprophagous on plants, but a few have apparently made transitions to either be saprophagous on animals or to feed on living plants, or in a couple of groups to attack other insects or vertebrate animals. So this is just to show the diversity of the modified ovoscaps within this clade. So there's not exactly a clear synapomorphy between these groups that have these modified female egg-laying devices, but nearly all the groups have one sort or another. So you can see within, for instance, the micropesids and the psyllids, there's different modifications, but they all are heavily modified in some sense. Within the tetraptoids, the major finding at the family level is that the polypterids are not monophyletic, and polypterids include both plant-feeding and parasitoid lineages. So even those apparently specialized ecological strategies are evolving independently. Here's an overall picture of the tetraptoids in terms of their life history traits. The green ones are plant-feeding. The red ones are predators or parasitoids, and the black ones are the saprophagus on plants. So we can see just over here, while I'd like to get more samples and do more analyses to get a more clear ancestral state reconstruction, is that plant feeding has evolved many different times, although within the tetraptoids it's the most diverse, and parasitoids have also evolved several different times. So overall, in the tetraptoids, we have a preponderance of evidence that the tetraptoids are sister to the clade flies that include Drosophila and the acalyptrates. And this has enlightened some novel biological trends in terms of the female egg-laying devices. Polypterity is probably not monophyletic, and phytophagy has evolved many different times within this clade, whereas it has evolved relatively few times in other groups of flies. So why has that happened? It's something that's quite interesting to consider. So within the tetraptoids particularly, the female ovis gap is usually extended during mating, and the male adyagus is extremely long and coiled. And this predates, this is in groups that evolved and split off earlier than the phytophagus groups. So it could be that we are considering the ovis gap as an egg-laying device, but perhaps it's also important in terms of mate choice and cryptic female choice during mating. So we are sampling a lot more tetraptoids, and we'll have a relatively complete picture of the phylogeny of this group. And I also want to get into the largest group of the largest family of acalyptrate flies, the tephridids. So tephridids are have a lot of lineages that have major economic importance, but have hundreds of other genera that are plant feeding, or some are saprophagus or parasitoids that are much more poorly studied and quite morphologically diverse. So I am a member of a large consortium of fruit fly workers worldwide, and we are trying to get together at least one representative of each genus of the family. So currently we have 32 out of 40 of the tribes and other family level groups, and almost half of the genera. So this is already surpassing the one previous molecular phylogeny of tetraptoids from 2016. We want to use this as a framework to test particularly these trait data in terms of how females find their hosts, and also host plant and host natural history relationships. So here's some of the network of collaborators as part of this study. And if anybody has experience and interest in tetraptoids and samples or expertise, please get in contact with us. We're trying to be as inclusive as possible. The major question within the tephridids is the monophyly of the family itself. So there's two other families, the ctenostylids and the pyrgodids. The pyrgodids are parasitoids, so they attack mainly scarab beetles, and the ctenostylids are likely parasitoid but have never been reared out. They have a lot of heavily modified adult morphology that suggests it's used for finding hosts. Another major group of the tephridids are the phytalmioines. So the phytalmioines tend to have very strong sexual dimorphism, where males will battle over mate choice for the females. And most phytalmioines are saprophagus. They're mostly in rotting plant material. And the blepharonurines is another major lineage that are plant feeding. So essentially what it looks like is the picture here in terms of both the monophyly tephridids and the ancestral states of different lineages of tephridids is quite unclear. So either parasitoids evolved twice within the plant feeding groups, or plant feeding groups evolved twice, or there's been a major reversal to saprophagy within the tephridids. So I'm very happy working on these beautiful and interesting flies, and I think it's going to be quite a challenge to both get sufficient sampling and really investigate what were the evolutionary historical patterns that led to the tephritid fruit flies becoming one of the most successful plant feeding fly lineages. And within that, the current classification in some cases doesn't capture the evolutionary history of the group. For instance, the tripetines. So that's a famous tripetine, Toxotripina now Anastrefa, with that extremely long ovoscap ovipositing into a mango. And again, the male of that has to have an ediogus almost equally as long to mate with the female. So that subfamily is not monophyletic with respect to the other subfamilies. And within the tephritines, those have mostly specialized on the flower heads of Asteraceae. But within the tephritines, there's a huge amount of genera and tribes that don't match the current phylogeny in terms of their monophyly. So this is really where this is going to go in terms of trying to understand where and when plant feeding and parasitoidism has evolved in this major group of plant feeding flies. Overall, with the tephridids, what I'd like you to take away is that the family is not monophyletic. Plant feeding blepharonurines and saprophagus phytalmioines are apparently closer to the parasitoid purgodids. Tachyniscines is another parasitoid lineage which we need to include. So just within tephridids, either plant feeding or parasitoids evolved twice, or there were major reversals. And tripetines are not monophyletic, and the major group in terms of genus and species level diversity, the tephritines, which has never been a very strong phylogeny. This approach of combining anchored hybrid enrichment, transcriptome, and genome data together is looking quite promising. So there's still a couple major taxa that we'd like to get our hands on. So if anybody is in Australia and would like to do some field work, or elsewhere in the world and has a lead on any of these taxa, please get in touch. We're very happy to share data and collaborate. And with that, I'd like to make some acknowledgements. Hopefully, have a couple of minutes for questions now. And thank everyone again for having me. And please stay on the line for Dr. Pittman. Thank you.